what do we have here?
Hey everyone, glad you could join us out here. It's nice and crisp. We have a great evening. If you'd like to bring the camera in here, Tommy, I'd like to show you guys our two kinds of beautiful base pairs, our four bases represented by the wonderful colors red, yellow, blue, and green. Now as you can see, the colors represented are bonded with each other right there. All right, now let's get, yes.
Okay.
Now let's get right into how DNA replicates itself and allows all biology to be beautiful. So first, we're gonna start out with the helicase and the topoisomerase. So what this does, this one here is the topoisomerase. It helps the helicase unbind the DNA. So right there. Now the reason we need this guy here is because sometimes the strands can be wound so tight that the helicase, this guy, can't do his job. So we separate here what is called the point of origin, right there. Excellent. Okay, so this is our point of origin here. This is our original DNA strand that was unwound by these two guys. Now, here we have the DNA polymerase 3 and the DNA primase. Now the polymerase 3 can't do his job without the help of the primase.
What was that? What was that? Did you hear that? Did you hear that?
All right, now what the primase does is it brings a RNA primer here on this leading strand and sets it there. So now we have what is right here. This is our RNA primer. Now the DNA polymerase 3 can do his job. Now he brings in this set of DNA. It doesn't come in like that, but he builds it onto there. And notice the colors are matching with their base pairs, just like in real DNA replication. So the polymerase 3 has done his job, and keep in mind this is going on this strand as well, utilizing things such as Okazaki fragments to what happens is they put down a primer, create it, another primer, create it, and that brings us to our next happy helper, DNA polymerase 1. Now DNA polymerase 1 converts the RNA primer into DNA. Look at that. Wow, it's beautiful. It's beautiful. And then DNA ligase, who's always following primase 1.
How many people know about this?
2. DNA ligase, who's always following DNA primase 1 around, comes in and he bonds these two with a phosphodiester bond. Right there. It links those two together because they were made differently. So you've linked those two together. Now I do want to note, the whole time this is going on, we have our single-stranded binding proteins holding the strands in place. If you can see those, these guys are representing our single-stranded binding proteins. So that's pretty exciting. Now we also have exonucleases running around, making sure that the DNA is staying in place properly. They're being held and they're working together. So you can see this beautiful replication here. We have a totally new strand that is almost identical to this one. Now, we can't move on without mentioning nucleoside triphosphates, which we're just gonna take this guy apart and represent with this. So, the nucleoside triphosphates make sure that the DNA was made properly.
Yeah.
Say, for example, he sees this. He sees any errors. Oh, fix that right there. Boom. Nucleoside triphosphate. All the work is done. So here we had our binding proteins holding it together. We have our DNA polymerase 1 turning the RNA primer strands into DNA. We have the ligase making phosphodiester bonds to create new strands. We have our primase, which is the one that creates the RNA primers, our helicase, our topoisomerase, and our DNA polymerase 3, which in my opinion is the coolest because he just does the bulk of the work in my opinion. So there we go, guys. Here's our new strand, and our old strand, don't worry, we're gonna build another one here in a minute. Thank you, everyone. Have a wonderful night. Drive safe in the winter. Amen. How'd I do?
You did all right.